So eukaryotic gene expression. One way is to determine and to control the chromatin itself. Is it physically able to be transcribed? In addition to that, we also have transcription factors. Transcription factors that bind to the DNA sequence and thereby affect its ability to be transcribed or not transcribed. Activator proteins are about as complex as it gets, my friends. Here is a transcription complex. Activator proteins that connect to the distal end or the far upstream end of a piece of DNA before a promoter Tata box gives it almost a scorpion tail kind of effect, comes back over across the top, and when these activators make contact with these other proteins, the RNA polymerase, the promoter complex for initiation. This initiation then of transcription can start. But notice all of the different activators and co-activators that are necessary here in order for this initiation complex to begin. Complexity doesn't begin to describe this, but imagine the number of controls in place. These activators are proteins, and any one of these proteins can be controlled allosterically. Any one of these proteins could be not transcribed from its DNA regulatory gene. So a hyper level of control in the eukaryotic organism. Here is the activator protein model bound to a DNA for some more detail. So you have these enhancers, either proximal or distal. These are the same kinds of terms that are used in anatomy. Proximal and distal control. Okay. Proximal being part of the promoter, distal being enhancers that would have an effect of that scorpion effect of connecting to the DNA. All of which allows for a more complex level of operon, if you will. So here's our activators way upstream that have to roll over. Okay, we have a DNA bending protein that's going to cause this to roll over, a group of mediator proteins in the middle here. And when all of this comes together, when that scorpion tail swings all the way over and strikes, now the RNA polymerase is able to go, and the initiation is going to happen, and RNA synthesis can begin from that point. So control at multiple levels. The nucleosome level control via methylation or acetylation. Are we going to cramp together that DNA or relax it so that it can be physically transcribed? And the pre-transcription kinds of controls due to the protein interactions of all of that initiator complex. Multiple levels of control for eukaryotic transcription.